going to get started today. Thank you all for coming. It's my pleasure to present Dr. Lawrenz. He's an associate professor in the microbiology and immunology department. He's also a member of the Center for Predictive Medicine and one of my colleagues who I collaborate with. So we're fortunate to have him speak today. A little bit of background about Dr. Lawrenz's history is that he obtained his Bachelor's of Science in Microbiology at Colorado State University in Fort Collins, Colorado. His PhD in Microbiology and Molecular Genetics at the University of Texas, Houston Health Science Center. And he completed 2 postdoctoral fellowships at Washington University School of Medicine in St. Louis, Missouri, and also at the University of North Carolina, Chapel Hill. He was an assistant professor, now an associate professor, and has a number of thorough publications and is very well funded in some of the work on defense and bacterial infections that he'll be talking with you about today. And I'm sure you'll all find it pretty interesting how we can look at things from the basic science level and then translate that into some of the things and concerns we see on the news in terms of pathogenesis and bacterial infections in the public eye, if you will. So we're grateful to him for giving a talk, and I'll let him get started.
Thanks, Joe. We might want to turn the lights down a little. There will be some fluorescence microscopy eventually. So, well, I appreciate you guys coming out to see a talk by, it's going to be very kind of basic microbiology, but hopefully I can get you guys interested in why we're interested in the disease known as plague. I scrolled some headlines here. As you guys can see, whenever there is a plague case in the U.S., it tends to trigger these type of headlines in the news. So, historically, when we think about plague, most people think about this in a historical fashion, and that makes a lot of sense because of these 2 major pandemics that occurred. The Justinian plague in the 6th century, which was responsible for about 100 million deaths, and it coincided really with the final downfall of the Roman Empire. And then the Black Death that began in the 14th century, which most of us know because of Ring Around the Rosies and all of the links of the Black Death in the medieval times. And really this led to kind of the collapse of the feudal system and allowed for the opening of the Renaissance. It's estimated conservatively that about 1/4 of the world's population was killed during the Black Death. You know, when we talk about the world's population, we're talking about the old world being Europe, Asia, and Africa. Some of these estimates have gone as high as 1/3 of the world's population died during this pandemic. Down here is a plague burial site, a mass burial site that was unearthed in London. They were digging up a tunnel for one of the subways and they came across this. And just to give you an idea of what was going on, during this outbreak in about 1350s in London, there were 800 people a day dying from the plague. And so the only way that they could handle this was these mass burial sites. And this is a plague doctor from the 1350s, and I just want to point out that they wore very important personal protective equipment, or PPE as we call it, which include these robes, a mask, and they had this stick that they would poke their patients with to try to protect themselves from this disease, even though they didn't understand how the disease was transmitted. What most people, maybe my remote doesn't work. Oh, there we go. Most people don't realize is that there was a 3rd pandemic that began in 1855. And if you guys are interested in epidemiology, this 3rd plague pandemic, there's great books out there studying the spread of it, beginning in Hong Kong and spreading all across the US. And this was at a peak time when microbiology was really coming into its own, starting to understand that bacteria cause disease. And it is at this time that this disease was recognized as being caused by a bacterial pathogen. This is an ongoing pandemic. We're still in this now, and we've seen over 12 million deaths worldwide. The one thing that I want to point out that this pandemic was a little unique from the previous 2, and this is the first time that this disease moved to the New World. And I'm just trying to highlight that here, focusing on the US, that's where we live. You can see here that these states in green all have plague endemic in rodent populations within those states. These yellow dots all represent human plague cases over this about 100-year period. And you can see that all of this is confined to the western United States at this point. But there's a reason for that. And the reason is that being an old world disease, it wasn't here until a ship showed up in San Francisco in March of 1900 and brought this organism on rats on that boat to San Francisco. They put a quarantine in place to protect, but the rats made it past that quarantine. And so now the disease has been steadily moving eastward. And there's no reason to believe that it isn't going to continue to move all the way out to the eastern seaboard. So eventually, we may have to worry about plague in Kentucky. I wanted to show you a plague doctor from the 2000s. This is how we work with plague. It's actually a very dangerous disease. It can be transmitted by aerosols, and we'll talk a little bit about that. But when we work with this, we work in what's called biosafety level 3 conditions, where there are a lot of engineering protections in place, negative airflow, working in biological safety cabinets. But we also wear our own form of PPE that's come a long way since here, although it looks very similar. We have a mask where the air is HEPA filtered. So we're breathing purified air. This blows over your face the whole time, so you're essentially under positive pressure. And all of that air is filtered. And we wear gowns, gloves, etc. to protect this from this organism. Now, as I alluded to a little bit, this disease is caused by a bacterium known as Yersinia pestis. It's a gram-negative bacterium. There's no vaccine for this disease. And while it is currently sensitive to antibiotic treatment, being a bacterial pathogen, we're always concerned about the development of resistance to antibiotics. And there have been a few isolates in Africa and Asia that have been isolated that do have resistance to antibiotics. These are typically found in the environment and are not typically isolated back from plague patients as of yet. But there's no reason, again, not to believe that this bacteria isn't going to follow the same trend as staph or Pseudomonas and be able to pick up these resistance to antimicrobials. The bacteria is a zoonotic pathogen. What that means is that you and I aren't the normal host for this. Really, this is a pathogen that infects animals. In the wild, it is maintained and infects primarily small rodents, and it's transmitted by an insect vector. So in the case of plague, it's transmitted by about 80 different species of fleas. And so you have this end-zootic cycle that's going on all the time in the wild, with a relatively resistant host having the organism being fed upon by a flea, transmitting to a new host, and this is the reservoir. This is where it's maintained in the environment. And it's actually very rare that we see transmission from these reservoir hosts to humans. The problem occurs when in the wild you get what's called an epizootic event, or essentially an epidemic of rodents or animals that are susceptible to this disease. In the US, we tend to see this in prairie dogs. If you're not from the western United States, prairie dogs are about yay tall. They're burrowing animals, and they form what are called prairie dog towns out west. And these prairie dog towns can have hundreds of animals within them. It turns out that these prairie dogs are extremely susceptible to plague. When it enters into a prairie dog town, it'll kill about 98% of the prairie dogs that are in that town. And the problem with that is you still have these fleas jumping around that can transmit the disease who are hungry. And so they go out looking for another host. And this is where we primarily see the transmission to the human host. And this can occur if you and I come in contact with this dead animal, the flea jumps off and takes a blood meal and transmits the organism. Or in the US, what we tend to see is our pets will go out and interact with these. And they'll bring the fleas home. Dogs or cats will bring those home. Most of us allow those animals to sleep in bed with us. Fleas will jump off the dogs or cats. And we get transmission in the US primarily through that route. Plague in general is a very acute infection. Individuals that come down with this can die within 7 days after exposure to the organism. And it has a very high mortality rate without proper treatment. Anywhere from 50 to 80% of individuals who don't receive proper treatment will die from this disease. Again, that goes back to those big pandemics that we saw in the historical sense. Again, it is treatable by antibiotics, and so most people that come down with what we call bubonic plague, if they receive medical treatment, are going to recover from this disease. Now, there is the possibility that if you don't receive treatment right away, this organism can move into the lungs. It can colonize the lungs, and it can cause what we call pneumonic plague. So you get the development of pneumonia, the bacteria replicate, and you can aerosolize this bacteria when you cough. And so one of the scary parts of this is that you can begin to see person-to-person transmission. So in the case of pneumonic plague, you can now aerosolize it. A naive individual can breathe it in, and they'll have a primary pneumonic plague case where the disease starts in the lungs. And you can see this with in the hospital. If individuals aren't known to have plague, your first-line responders can interact with them and come down with primary pneumonic plague. Again, this is an acute infection. In this case, death can occur as little as 3 days. And the mortality rate is 100% without antibiotics. And it's actually pretty close to 100% even with antibiotics. And that's because by the time you develop symptoms of pneumonia, it's usually too far along for antibiotics to be helpful to the patient. There was an outbreak, I don't know if you guys saw it on the news, but there was a big scare in Madagascar kind of over Christmas time, maybe right before Christmas, where they had about 2,000 individuals in Madagascar came down with plague. Madagascar always has plague, but they tend to have this form, bubonic plague. And in this outbreak actually occurred in a major metropolitan area, and it was primarily a pneumonic plague infection. And so they were getting that person-to-person transmission. And this was very reminiscent of the Ebola outbreak that we had about 2 or 3 years ago, where people started getting concerned about those individuals getting on planes, going to other countries, and carrying plague with them when they're on the planes. We were very lucky here. There was no transmission outside of Madagascar. Out of those 2,000 people, about 10% of them died from this infection. They were able to bring that under control, and that epidemic has died down at this point. The other concern here is that when you have that aerosol transmission route, is that this organism could be weaponized and used as a bioterrorism agent. And in fact, it has a history of being used as a bioweapon. And so for that case, work with this organism is highly regulated by the federal government. It's considered a Class A select agent and requires us to go through a lot of regulations with the CDC in order to work with this organism. Now what I want to highlight to you guys one more time is the fact that this is a very acute infection. And if you think about this from an immune response standpoint, the short time that this infection takes place before the host dies means that the only arm of the immune system that the organism really has to deal with is the innate immune system. So that your first responders, your monocytes, your neutrophils, and not so much with the adaptive response. Now the adaptive response is important to resolve the infection, but all of the evolutionary pressure put on the pathogen is really driven by these innate immune cells. And so because of that, my laboratory primarily focuses on trying to better understand the interactions of Yersinia pestis with the innate immune system. And the work of my lab is really broken into 3 different parts. A lot of effort by several students in my lab is focused on specifically those interactions with those first-line responder cells. And so we're interested in how does Yersinia pestis survive within macrophages, and how do they subvert the normal killing mechanisms of neutrophils. We also have a relatively new project in my laboratory that's focused on nutritional immunity. And nutritional immunity is our ability to restrict nutrients from pathogens. It's one of the first responses that we make during a bacterial infection. And one of the big ways that we do this is we restrict transition metals like iron, zinc, and manganese in different mechanisms so that it's not readily available for bacteria to use. All bacteria need metals, actually everybody, every life form on Earth needs metals in order to grow. And so if we can restrict those, it actually works pretty well at restricting normal bacteria from replicating within the host. Pathogens have got around this by coming up with different mechanisms to acquire metals. And one of these is to make small compounds that have extremely high affinity for these metals and can outcompete those restriction mechanisms or sequestration mechanisms by the host. And so we're interested primarily in how Yersinia is able to get zinc away from the host and be able to use that in order to grow. Now ultimately, both of these projects are really focused on a basic understanding of the biology of this organism. But we're hoping that we can use that basic biology to translate that into ways that we can combat this infection. And primarily, we have active work going on in the lab looking at this idea of immunomodulation, or modulating the immune response to be favorable against the bacteria and provide protection. And we do this by a couple different ways. We have a collaboration with UofL to try to develop new adjuvants for vaccines. And adjuvants are the part of the vaccine that stimulates the immune system. The antigen is what gets targeted by the immune system. The adjuvant is there to stimulate that immune response. And then we're also interested in ways that we can manipulate these cells, either neutrophils or macrophages, to be better capable of killing the bacteria. And in this case, we're using nanoparticles or microparticles as drug delivery mechanisms to deliver molecules that activate these cells and make them better capable of killing. And again, all of that is based on how, once we understand what's going on here, we can figure out ways to manipulate those cells to be more capable of killing the bacteria. Now, what I'm going to tell you guys about is a story that we published, I guess, our second major paper on just earlier this year, looking at how does Yersinia pestis survive within macrophages. And the majority of this work was done by a very talented PhD in my lab, Michael Connor. So a lot of what I'm going to show you today is all from his thesis. As I said, this ability to interact with the immune system means that that's where a lot of the pressure is coming on the bacteria from an evolutionary standpoint. And so the bacteria itself has evolved many different virulence mechanisms that allow it to evade the innate immune system. And I just want to give you a couple of examples here to try to drive home different strategies that are used by this bacteria. So one of those is that it alters a molecule in its outer membrane called LPS. So all gram-negative bacteria on their outer membrane have LPS. Because of that, the innate immune system has evolved to recognize that as a foreign antigen. And so through a receptor called TLR4, it will recognize LPS and that will stimulate a pro-inflammatory response. The bacteria, in case of Yersinia pestis, has actually evolved to change its LPS. Normally, these are fatty acid side chains here in blue. Normally, LPS has 6 of those, and that's what's recognized by the immune system. Pestis changes its LPS so it only has 4. And by doing that, it's no longer recognized as a foreign molecule and doesn't stimulate the immune system. The bacteria also makes a capsule, and I'm trying to show that here. This is an EM of the bacteria. You can see these long filaments coming off that are called CAP1 filaments or CAP1 capsule. So this is a cloud around the bacteria, and this protects the bacteria from being phagocytosed by macrophages and neutrophils. And then this third one I'm showing you here is probably the most important virulence factor for Yersinia pestis. It's a type 3 secretion system. Essentially what that is it's a machine that's made by the bacteria. So this is the inner membrane of the bacteria, outer membrane of the bacteria, and the host cell membrane. And so this is essentially a needle that the bacteria produces that allows it to inject proteins directly from inside the bacterium to inside the host cell. And once those proteins enter into the host cell, they're going to mess up normal signaling pathways within that host cell. They're essentially kind of like toxins. In the case of Yersinia pestis, it injects only 7 proteins into these host cells. But what that does is it inhibits phagocytosis by neutrophils and macrophages, so they can't eat them. They will induce cell death in those cells so that they're no longer there. And they inhibit that pro-inflammatory immune response to recruit more immune cells into the site of infection. Now, the dirty little secret in the Yersinia field is that this organism's in two different hosts, right? The mammalian host, you and I, and the flea. And it turns out that in the flea, it doesn't need any of the virulence determinants. Those are specific for the mammalian host. And so they don't express those in the flea. And that means right upon transmission from the flea into the host, there's a period of time when the bacteria are very susceptible to phagocytosis by neutrophils and macrophages. And I hope you guys can see this up there. What I'm going to show you, this is intravital microscopy looking at the ear of a live mouse that was infected with Yersinia pestis. In red is going to be the bacteria. It's also a transgenic mouse where the neutrophils and macrophages express green fluorescent protein. The neutrophils are going to be bright green like this is a neutrophil, and the macrophages are going to be a more dim color. And so I'm going to start this, and I hope you guys can see it. But I just want to highlight what's going on at this time of infection. That arrow, and you can't see it, but it's right here coming down, is a macrophage that took up one of these bacteria. And it's actually in there and is not degraded. And what you can see is that eventually these neutrophils recognize those extracellular bacteria. They come, swarm in, but they pay no attention to that macrophage that has an intact bacteria within it 4 hours later. Normally, a macrophage will degrade a bacteria in about 60 to 80 minutes. 4 hours later, that bacteria is still there and not recognized by those neutrophils. And if we, again, take another look at this, now this is looking in vitro, looking at infection of peritoneal macrophage. The macrophage is going to be kind of outlined here. The bacteria are in red here. And so this macrophage has been infected with the bacteria. And this is going to be a 24-hour time point. And you'll see real quickly here that the bacteria aren't being degraded. The macrophage is moving around, appears healthy. It's now rounded up. It's lysed. And you can see that all those bacteria are viable. They come out into the media. They just expand and grow. What I like about this video is we caught this event where this second macrophage has come up, investigated the area, and you saw that it wasn't phagocytosed by the bacteria weren't phagocytosed by that macrophage. And that's because when it came out of the back end of that first macrophage, it turned on that type 3 secretion system. So then it interacted here, and you see that blockage of phagocytosis, and actually killed it, rounding up, induced apoptosis, and killed that cell. We can monitor this by different ways. This is by microscopy. This is using bioluminescence, so we have a strain of Yersinia that makes light, and we can monitor over time, and you can see that these are all intracellular bacteria over a 24-hour period, and you can see replication. If we take a closer look now inside that macrophage, we can see we try really hard to understand why this macrophage isn't killing the bacteria. This is a super infected macrophage, about 18 hours post-infection, where this macrophage has been infected with multiple bacteria. But what I'm trying to highlight here is that every single one of those is inside its own compartment within the macrophage. So it's not just in the cytosol, it's actually within a vacuolar compartment that we call the Yersinia-containing vacuole. And up here, you can see now the bacteria have begun to replicate within that vacuole. And what happens is they're still in that vacuole. The vacuole is expanded, and we call this a spacious vacuole. There's an important step that goes on normally during macrophage phagocytosis. If the macrophage was going to eat a non-pathogenic bacterium, what normally happens is you have phagocytosis into a phagosome. That phagosome will acidify. That acidification then leads to a signal to fuse with the lysosome, and the lysosome will then dump antimicrobial components into that phagolysosomal compartment. And that leads to how the bacteria are degraded. In the case of Yersinia, when we look at these vacuoles, almost none of them are acidified. This is compared to a PFA-killed Yersinia pestis. You can see that almost 90% of them are acidified in as little as, I think this is 20 minutes post-infection. But the Yersinia is able to inhibit that acidification. So a key step into how the macrophage is able to degrade it. That also translates into fusion with the lysosome. LAMP1 is a marker for lysosomal fusion, and you can see in the case of the PFA-killed bacteria that by 80 minutes post-infection, we're already up to about 60% have fused with the lysosome and have those antimicrobial components, where the live bacteria really inhibits this. And so essentially, what's happening is that the bacteria is subverting phagosome maturation and making a replicative niche within those macrophages. Now there's, I think, pretty good evidence that this ability to inhibit killing by macrophages contributes to the overall virulence of the organism. We've known since the 1950s that you can isolate viable intracellular bacteria from not only animals infected with plague, but also patients that have plague. In 2009, over at University of Kentucky, just down the road, there was a great study that came out of Sue Straley's group where they depleted macrophages and dendritic cells from animals and then infected those with plague, and actually saw that the animals that were depleted of those host cells did better than the ones that had macrophages, indicating that perhaps those macrophages were providing a way to hide from the immune system during those early stages of infection. We also know that mutants that are attenuated for intracellular survival in cell culture are also attenuated in animal models. More recently, there's been a couple studies, one with dogs and one with a resistant rodent line, that showed that the macrophages from those animals, and those animals are resistant to plague normally, their macrophages are much more capable of killing the bacteria than our macrophages or macrophages from laboratory mice. And then finally, there was a study out of Skip Virgin's lab at Washington University that showed animals that are infected with herpes have kind of an elevated immune response going on all the time because your immune system is fighting that chronic infection of herpes. And what that means is actually your people, in this case animals with herpes, their macrophages are just a little more activated than normal. And it turns out that that kind of background higher activation of macrophages protected those animals from infection with Yersinia. So when you bring that all together, this really argues that this ability to survive within macrophages is an important contribution to overall fitness and virulence of this organism. So our kind of overall hypothesis here is that the organism produces virulence factors of some nature that specifically interfere with the ability of those macrophages to kill by the normal phagosome-lysosome pathway. For the last 5 years or so, we've been working towards the goal of trying to identify the factors made by the bacteria that allow this to happen, and also define the molecular mechanisms that lead to this subversion of the innate immune system. Now, the traditional approach to do this is to target the bacterium. Essentially, you make a mutant in the bacterium that can't survive within the macrophage, and then you use that mutant to understand the mechanism that is involved in that evasion of killing. The problem with this is that, at least in the case of Yersinia pestis, conventional bacterial mutagenesis approaches haven't identified any virulence factors that really change the generation of that Yersinia-containing vacuole or YCV. And there's probably good reasons for this. The most important is probably this idea of redundancy. When bacteria interact with a host to avoid some signaling pathway, they usually make more than one factor that interferes with this. And so conventional mutagenesis approaches only allow you to target one factor at a time. So if there's a second factor that also contributes to this, many times you won't be able to find them because you don't get two mutants at one time. And all of this work was done by a variety of different labs. Gimbliscas, Australias, etc. And really hadn't yielded anything. And that's when Michael joined the lab. Michael was very interested in this phenotype, but he didn't want to take those same approaches that were done in the past. And so he thought that perhaps an alternative approach would be to target the host. And by targeting the host, what he wanted to do was identify host factors inside the macrophage that are required for the bacteria to survive. And the idea is that these are the signaling pathways that bacteria targets, and by identifying those pathways, we could then work our way backwards and identify the bacterial proteins that target those signaling pathways. So to do this, Michael set up a high throughput screening system where he took macrophages and he used an approach called RNAi. Essentially, we transfected these macrophages with a small RNA, which inhibits the expression of different messenger RNAs. And so you can do this and essentially knock down the expression of any gene within a host cell. He took those RNAi-treated macrophages, he infected those with Yersinia pestis, and then he looked for whether or not the bacteria was able to survive in the macrophage or not. And the phenotypes that he used, again, we used our bioluminescent strains so we could kinetically monitor. If this is the wild type situation, what he was looking for were phenotypes like this, where the luminescence went down, indicating that the bacteria were killed. Using this, he performed this high throughput screen where he targeted almost 17,000 genes individually within these macrophages. And then he performed a series of validation screens that included validating with individual siRNAs, but also challenging these with two different strains of Yersinia. One strain that lacked that type 3 secretion system that I told you about, and one strain that had that type 3 secretion system. And a long story short, what Michael came down to when we started after the validation and running it through bioinformatics was that there were 55 genes that he felt very strongly had some contribution to the survival of Yersinia pestis within those macrophages. And we can look at this different ways by bioinformatics. I'm not going to show you all. I'm going to show you a very simple kind of gene ontology way to look at this. But there were three groups of proteins that really popped up to us. Those involved in vesicular-mediated trafficking, and that made sense. Remember, Yersinia is inside a vacuole. Regulation in response to stress, of course, being infected by bacteria is a stressful response. And then this third group, which I'm going to focus the rest of today's talk on, are these small GDPases. And primarily what the GTPases that Michael was interested in were in a family called RAB GTPases. And RAB GTPases are central mediators of vesicular trafficking within the host cell. They pretty much govern all of the transport of any vesicle within the host cell. When we look at this from, I guess, as a bacteriologist looking at interactions with macrophage, we tend to look at it again in this phagosome maturation process where you get acidification, phagocytosis, acidification, which then leads to fusion with the lysosome and degrading of those contents. And this is all mediated by a series of RAB proteins. RAB 5, 7, and 9 are important in this. What Michael saw when he enriched for factors that were required, again, these are host cell proteins that are required for the bacteria to survive. What he saw was that he got an enrichment for several RAB proteins that are linked to the recycling pathway. And this was very interesting to us because when we think about endocytosis and phagocytosis, while this is the normal pathway that we think about as a bacteriologist, there's a very important step that takes place in this sorting endosome where you can either move down the phagolysosomal maturation pathway or you can enter into the recycling pathway. And this is used by the host cell to reuse receptors on the plasma membrane of the cell. And this is mediated by a different set of RABs, RAB 4 and RAB 11. So I'll just show you some of the kind of raw data here. This is looking at knockdown of those host proteins, and we looked at 4 in this case. RAB4 and RAB11 are those central mediators. And then we looked at 2 myosin proteins. And myosins are the motor proteins that bind to those RAB proteins and allow the vesicles to move. That's what gives the energy to move the vesicles to different parts of the host cell. So you can see here, this is just the RNAi, where we're looking at expression of those proteins, and you can see that the RNAi, we get at least 50% knockdown of all of these. Decreasing the expression that far had no impact on the viability of the macrophage. The macrophages were completely viable. But when you look at survival of Yersinia pestis, you see that in scrambled is our normal control. When we knock down any of these 4 proteins, you see a significant decrease in the number of bacteria that are present within those macrophages. So the way that RAB proteins work is that they actually integrate into the vacuole or the vesicle that they're going to transport. And so this is a very simplistic look at RAB biology. In the host cell, as I said, this is a GTPase, so the RABs exist in 2 forms. One bound to GDP, where it's inactive and in the cytosol. And then when they are activated by a GEF protein, you'll get binding to GTP, conformational change, and integration into that vesicle that they're going to transport. And since we hit these in our screens, actually one of Michael's first questions was, do these RAB proteins actually get recruited to that Yersinia-containing vacuole? And to do that, we used confocal microscopy, where we had RFP, red fluorescent protein labeled bacteria, and GFP, green fluorescent protein labeled RAB proteins. We infected the macrophages that were expressing these GFP proteins, and then we looked at whether or not the signal co-localized with the bacteria, indicating recruitment to the YCB. And what you can see here on the top is RAB4A, and on the bottom is RAB11B. And you can see that when macrophages are infected with live Yersinia pestis, in both cases we get a high degree of co-localization, indicating that those proteins are directly recruited to the YCB. Especially if we compare that to macrophages fed either killed Yersinia pestis or E. coli K12, which is a non-pathogenic bacteria. Both of these end up in the phagocytosis compartment and are degraded by the macrophage. Now, if we look over a longer period of time, we begin to see some differences between RAB4 and RAB11. You can see that in the case of RAB4, over about a 10-hour period, we have early recruitment, where the RAB GTPase is lost over time. But in the case of RAB11, we see that the protein is retained over the entire course of infection at the YCB. So it's essentially sequestered to the YCB. Now, as I told you guys, we knew before we got into these studies that the bacteria is very capable at interfering with this phagosome maturation process. And the phenotypes that we tend to look at are acidification and fusion with the lysosome. And so Michael wanted to know if these proteins are required to inhibit that maturation process. So again, live bacteria, the YCB very rarely becomes acidified. In the case of PFA killed, now it is acidified at a very high rate. What he did here was use that RNAi again, and then used a combination of fluorescent bacteria with a dye called lysotractor, which accumulates in an acidified vacuole to monitor acidification of the YCB. Now, the data is shown here. Killed bacteria normally are acidified. That's repeating what's up here. Scrambled siRNA is our control. That's our wild type situation. And you can see that in as little as 20 minutes post-infection, if we inhibit RAB4A, almost all of the live bacteria end up in acidified vacuoles. So this protein is absolutely essential to avoid acidification. When we look at RAB11 and myosin 5B, we don't see much of a contribution at 20 minutes. There is a slight contribution at 80 minutes, but it definitely does not contribute as much as RAB4 does. And this impact of acidification with RAB4 translates again into fusion with the lysosome, where when we inhibit 4A, essentially it looks just like killed bacteria, unable to avoid fusion with the lysosome. All right, for the last kind of 5 or 10 minutes here, I just want to shift gears a little bit. And what I want to tell you guys is that when we saw this involvement with the recycling endosomes, this is the first time that the bacteria had ever been linked to the recycling system. And so because of that, we were curious about what's the actual impact of infection on macrophages and on the global recycling that takes place by these cells. So again, recycling is this mechanism used by host cells when they have receptors that bind to a ligand. They get, this will induce a signal, right? And eventually you want to turn that signal off. And the way that the host cell does that is through endocytosis. In the endosome, you get removal of that ligand. This then recycles back to the plasma membrane, allowing reuse of that membrane protein or receptor to go about its business along. So this is a way to reuse components of the host cell. To monitor recycling, we used a receptor called transferrin receptor. This is a well-studied receptor in recycling. And the beauty of this is that you can use an antibody that will bind to this receptor. It won't interfere with ligand binding, and it won't interfere with endocytosis or recycling. So essentially, it hitchhikes through this whole process. And what you can do is you take a cell, you incubate it live with the antibody. It will go through the recycling, and you can then treat this with a buffer that will remove the extracellular antibody. And what happens is that you get the accumulation of only intracellular antibody, and if you have a disruption in recycling, you see more antibody inside the cell. So what Michael did was he took these cells, treated them with this antibody, and then infected them with Yersinia pestis, and just asked, does this change the ability of recycling with the transferrin receptor? And what you can see is that it absolutely did. This is just a dose response curve where you have uninfected cells. This is your normal recycling rates. And as you increase the number of bacteria, you get an increase in accumulation of these endosomes, indicating disruption in recycling. The important part here is that this is an active process taken out by Yersinia. If you have killed there, you don't see it. It's specific to Yersinia and not to other bacteria, where E. coli here is, if you put E. coli on here, you don't see a change in recycling. So this isn't just a normal response by macrophages. And if you add another pathogen, in this case Salmonella, you don't see that same thing. So it's specific to Yersinia pestis. The question that Michael had was that, does this result, how does this inhibit recycling? So the idea he had was that this was all dependent on sequestration of RAB11 on that YCB. Where you have, if this is your normal pool of RAB11 in the cell, when you have an infection, you're sequestered here and you have a depleted pool of the proteins that are important for recycling. So we thought he could test this in a very simple way, which was by overexpressing RAB11 and seeing if he could restore the ability of the cells to go through recycling. And that's exactly what he saw. By simply just overexpressing RAB11, he could restore that normal rate of recycling within the host cells. More importantly, he took this a step further and asked if he restored recycling, would this actually impact pestis survival? And when he looked now at bacterial replication within those host cells, you can see here this is your normal bacteria. These are, normal macrophages, overexpressed macrophages. You can see that at 2 hours there's no difference, but by 24 hours what happens is, under the normal circumstance, the bacteria replicate, where the bacteria are unable to replicate when RAB11 is overexpressed. So if we pull all this together, the work that Michael did, I think actually contributed to a better understanding of what's going on inside that macrophage. and really identified kind of this two-step process that we think is going on within the host cell. The first is this idea of avoiding pathogen maturation. And this is very dependent on RAB4 and RAB1B, which I didn't tell you guys about, but we published this a couple years ago. These two RAB proteins are important for inhibiting fusion with the lysosome. RAB11, on the other hand, isn't as important for this inhibition of fusion, but contributes to the second step of infection, which is bacterial replication. And so through what we think is sequestration of RAB11, we get an inhibition of recycling, and then through mechanisms that we don't understand, that change in recycling leads to replication of the bacteria. Now, of course, we have outstanding questions here. We don't understand how this is contributing to bacterial replication. We have some ideas. We think that it has some link to autophagy, but right now we're still exploring that. And then ultimately, we haven't got back to that main goal that I told you guys about at the beginning, which was to identify the bacterial factors that are involved. We know it's not dependent on that type 3 secretion system I introduced you guys to, but we now have targets that we can use to go fishing for these bacterial proteins, using these as potential immunoprecipitation, et cetera, to identify bacterial proteins that interact with these host proteins along the way. So with that, I'm just going to quickly acknowledge the people that contributed to this work. All of this, again, this is Michael. He's now off at the Institut Pasteur doing a postdoc. He did the majority of this work, but there was a lot of help from Amanda Pulsifer here, and she's taken this over and is now trying to identify those bacterial factors that might target these proteins. We have a lot of collaborators here at the university and at other universities, and of course, knowledge funding from both UofL and the NIH for this work. So I appreciate you guys hanging out with all the pizza smell in the back. I'm happy to answer any questions, but again, I'm glad you guys could come out and learn a little bit about plague. And I'm happy to take questions if you guys have questions.